Creativity is thinking up new things. Innovation is doing new things. In the world of 3D innovation, each one's focus is on designing. Let's take care of modeling. Let's give your vision a 3D walkthrough. Let's collaborate together to coordinate better. Good morning, everyone. Myself, Dr. Sakshi Bharadwaj, Application Scientist, Elton Technologies, Bangalore. On behalf of Elton Technologies, I would like to extend my warmest welcome to all my viewers for joining us today and much obliged to have you all here for this consequent session on in silico strategies for designing novel molecules, the approach of 3D QSAR pharmacophore modeling and docking. A chemical structure, as we all know, is crucial in demonstrating biological action against a protein structure, right? Or I could say, in the intricate dance of molecular design, the 3D QSAR pharmacophore is the choreographer, revealing the meticulous interplay of chemical structures and functional group, guiding us towards the a symphony of novel molecules with precision and therapeutic promise. Pharmacophores are the 3D space or core for the biological activity of a molecule. The generation of 3D pharmacophore models or QSAR models needs some set of, I would say, the training set of data to train our algorithms and test set of data to validate our models. Hold on, it seems to be interesting, right? Let's make it more interesting and knowledgeable with our speaker of the day. I feel happy and honored to introduce our speaker of the day, Dr. Ekta Singh, Associate Professor, Department of Pharmaceutical Chemistry, Acharya B.M. Reddy College of Pharmacy, Bangalore. She has done her M.Pharm from BITS Mishra, Ranchi, and PhD from Rajiv Gandhi University of Health Sciences, Bangalore. She has more than 17 years of teaching and research experience. She has worked with investigational drug services of Yerle New Haven Hospital in the USA and Research Pharmacy, VA Healthcare USA. She has guided many undergraduates and the postgraduate students for their research projects. She has guided many students under the summer research training program organized by CSIR, Government of India during COVID pandemic. Furthermore, she has received grants from the university for her work too. She has more than 15 national and two international research articles published in scientific journals. She has her research interest in synthetic chemistry, green chemistry, drug designing, software for the molecular modeling, and peptide synthesis. Today, she will be highlighting the journey starting from the SAR to the 3D QSAR, 3D QSAR pharmacophore to the docking and the simulation. I welcome you, ma'am. I'm sure my viewers will absorb and learn the concept behind these techniques. And certainly, we will be taking all the questions at the end of the webinar. So please feel free to type your queries into the chat box. And now, without further ado, let's start it. Up to you, ma'am.
Thank you, Dr. Sakshi, for such a wonderful introduction. I welcome all the participants of today's webinar, and I welcome you all for the discussion on in silico strategies for designing novel molecules, the approach of 3D QSAR pharmacophore modeling and docking. Let us go to the small disclaimer that this webinar is only for educational purpose and we'll be using it for discussion on the possibilities of in silico designing and focusing on 3D QSAR. The rights of the content of presentation belongs to speaker and the Alton Technologies. Let's move on to the content of today's discussion. I'll be highlighting the concepts of structure-activity relationship, quantitative structure-activity relationship, 3D QSAR, 3D QSAR pharmacophore, docking, MD simulation, and a short case study how we can use all these concepts in designing new molecules. As we see that the drug discovery and its timeline takes lots of effort, money, lives, and the duration required for getting one active molecule, one drug molecule approved by FDA is a long journey for any molecule. So here we see when we start with almost 15,000 molecules, we reach upon one successful drug molecule. In the journey of drug discovery, when we start with, and on an average, this is the data which is derived on the average study and average success of a molecule which reaches to the FDA approval. So we spend approximately 6.5 years in preclinical studies, preclinical studies approximately 7 years, and clinical studies takes approximately 1.5 years. Then it reaches to FDA review. If we see the next slide, we'll be able to understand why it is important for us to have a strategy for drug design. To cut short the duration of the timeline of a drug discovery for a one drug molecule to become success, we can reduce the duration. And the strategy we require is de novo drug design. De novo is a Latin term which means starting from the beginning, anew. Why do we need to start from a scratch to make the molecule successful in a shorter duration of time? So there are some known ways in which drug designing can be done. When we see the primary method, previously when in silico studies were not there, structure activity relationship was being studied. So molecule was synthesized and it had to be done preclinical studies and activity has to be established. Based on its activity, a molecule was categorized good, bad, or moderately active. Based on all the information, the structure activity relationship was derived. So it was made for a series of molecules. And activity was considered to be cumulative effect. So when we have the structure activity relationship, it was about molecules in a series which were showing same activity in good, moderate, or low quality. When we consider QSAR, it is slightly different from structure activity relationship, where the contribution of each parameter, each descriptor, or each of the feature, physicochemical feature, it was quantified. On your screen is a typical example of structure activity relationship where we have a scaffold in the center, and when we change the functional group, the activity either increases or decreases. In comparison to QSAR, we can see in the next slide that both SAR and QSAR provide valuable information in drug discovery. Based on these information, we can design a new molecule. Both SAR and QSAR can be used, but SAR correlates the net effect of substituent, whether the molecule becomes active, moderately active, or less active on substitution. So this was the focus for SAR. Whereas for QSAR, we derive a contribution of one or more physicochemical properties independently or collectively. So when we say that one of the physicochemical properties is contributing towards activity, in what extent it can be quantified, in what extent it is able to increase the activity. So it can be plotted in X and Y axis. On your screen, you are able to see SAR versus QSAR, where SAR you have seen a photo where a collective effect of substituent on activity was shown. Here on your screen, you are able to see one of the linear relation of quantitative structure activity relationship. So in this graph, we can see that activity and effect of substituent can be correlated. This is not the case in many of the studies, QSAR studies, where the relationship is not linear. On your screen, you can just understand the difference of SAR and QSAR. So QSAR representation can be done in a graphical manner. So when we see the use of QSAR for drug design, prediction started using QSAR approximately 200 years. You might not have thought about the data, but it is almost 200 years since when we have started using QSAR. And the correlation between the properties, individual properties, dates back to approximately 150 years. So we'll see the Hans equation was derived for making a prediction. So QSAR was used for estimating the properties and the chemical properties of a molecule, and the contribution of each of the substituent, each of the physicochemical property contributed by one of the substituent is quantified. So we'll move on to the next slide, where we will understand that not only prediction of the activity, but also for understanding the hazards of the chemicals can also be predicted. So QSAR is not only for predicting the activity, also for the harmful effect, hazards, toxicity can also be predicted. So QSAR works as a main tool for reducing time, reducing effort, money, lives of animals involved in the drug discovery. Next slide, please. So application of QSAR continues when we correlate the physicochemical properties. And if it holds true that increasing the, for example, increasing the lipophilicity increases bioavailability in central nervous system. This is common understanding. If this relationship holds true, then we get a linear graph. In some cases, this correlation of physicochemical property and activity is not linear. Sometimes some deviation happens. Not only one physicochemical property, other properties are also contributing. In that case, we get non-linear graph. For that, we have different strategy for finding out how it works. So QSAR allows us to select the analogs which have good activity. When we do QSAR, we get the range of molecules which are good, moderate, or less active. And based on this information, new molecule can be predicted. New molecules' activity can be predicted. In this journey of study, if we find out any molecule which is showing activity and is not falling into 3DQSAR equation, this gives the idea that some other property has been contributing towards biological activity, which is not included in our QSAR consideration. So QSAR models are used for not only designing the containers, also for further improvisation of QSAR, including the the molecules which show activity and which do not fall into QSAR equation. For any QSAR model, as you see on the screen, we are comparing the physicochemical properties, which are also known as descriptors. The physicochemical properties are different types of contributing features. What we say features, for example, hydrophobicity, lipophilicity, electronic factor, steric factor, etc. The physicochemical properties can be of different types: 1D, 2D, 3D descriptors. The 1D descriptors are usually the concrete values, for example, molecular weight, density, melting point. So these are 1D descriptors. 2D descriptors are hydrogen bonding, polarity, dipole moment. So these are 2D descriptors. And 3D descriptors are three-dimensional. That means contribution of the feature as a whole, for example, volume, for example, steric hindrance, steric factor, hydrophobicity of the molecule. So these are 3D descriptors. There is a term used, fingerprint descriptors, in drug discovery. Amongst all the descriptors, which one is contributing most is termed as fingerprint descriptors. Usually it is more than one. So the Hans analysis, Hans and Fujita, they concluded an equation where they have compiled three descriptors. Log 1 by C is the biological activity. So biological activity is related to three descriptors and a constant, where log P is partition coefficient. So partition coefficient with K1, a constant associated with plus a constant multiplied with ionic factor and a constant multiplied with steric factor. So here log P, Hammett constant and steric factor, these three are the main contributors for the biological activity. They have been quantified in a equation, and K4 symbolizes other factors which might affect the biological activity. Now we see that when these three parameters are affecting biological activity, the hydrophobicity or log P, that is partition coefficient, it can be expressed in two different types. Hydrophobicity can be expressed in two different forms, in the form of partition coefficient and hydrophobicity constant. Hydrophobicity constant is the contribution of a functional group. Suppose we have a standard drug which has partition coefficient 3, and then we have removed the functional group and introduced ethyl group. Now the hydrophobicity constant of ethyl group on that scaffold is the partition coefficient of the design molecule divided by the partition coefficient of the standard drug. So these are used for quantifying the contribution of hydrophobicity. Let us see the effect of electronic effect. The Hammett substitution constant, A, alpha, symbols are there for that. It measures the electron withdrawing or electron donating ability of the substituent. So it is electronic effect of the substituent. So these substituents which are present on the scaffold, they contribute not individually, collectively for the biological activity. The third one which we will be discussing is steric factor. So steric factor is the volume, the amount of the steric space occupied by the functional group. Along with steric factor, there is molar refractivity that also represents the volume occupied by an atom, and Verloop's steric parameter, that is the parameter which we consider for, that is again a contribution made by the volume of the functional group. So it covers the van der Waals radii, bond length, and the bond angle. So these are also included in the steric factor. So basically, when QSAR equation was coined, these three physicochemical properties were considered. Later on, when QSAR was developed to 3DQSAR, more physicochemical properties, more descriptors, other contributing factors which were ignored in QSAR were included. So, though QSAR is extensive study of all the substituent and the net effect on biological activity, there is a possibility of missing out some of the important contributors of the biological activity, which is included in 3DQSAR. So in 3DQSAR, we do a comparative study of all the parameters along with their individual contribution towards biological activity. For example, how individually hydrophobicity affects biological activity, how individually ionization or splitting, accepting or donating proton affects biological activity. So 3DQSAR is more holistic. There are even further modified ways which we will be discussing in the due course. So in silico models are available for both QSAR and 3DQSAR, but 3DQSAR is able to cover a larger extent of factors which affect biological activity. On your screen, you are able to see an example of QSAR. It's a Bayesian model. This is a result obtained in BioVia Discovery Studio for a training set. For one of the training set, we got that there is a good contributor and there is a bad contributor. So when we see the functional group, we see in 3DQSAR, in QSAR model, we get two types of contributors. The larger the training set, you get a large amount of data which divides good or bad contributors. In the next slide, there is another example of QSAR, partial least square method. This also provides with information related to biological activity and correlation with biological activity. Still, these representation, these information remain incomplete. Let us see an example of 3DQSAR. How 3DQSAR we are considering to be better. With a pictorial representation in the next slide, we are able to see that in 3DQSAR, more information is involved. So in the next slide, we are seeing that in BioVia Discovery Studio, we can have the study of van der Waals force, electronic grid. So these two, if we consider for our training set and the effective, its contribution to biological activity, we can get a graphical representation also, and in 3D space, how these features, van der Waals interaction and electrostatic interaction contributes to biological activity can be represented. In the first one, what you see as yellow grid, it is van der Waals grid, and green dots where you see green patches, these are the positive interaction, and yellow patches are negative interaction. For a training set of 60 molecules, we found that this representation shows that those molecules which have yellow bar, they are less active. And let us see the other one where electrostatic potential grid is there, electrostatic interaction where ionization is happening. We see that blue is positive interaction and red is negative interaction for a training set. So from 2D interaction to 3D interaction, we are in an advanced phase of making a model for designing drugs. So QSAR is now obsolete. 3D QSAR is a better version. Now we have even more advanced way of in silico drug designing. So now we will discuss about 3D QSAR and 3D QSAR Pharmacophore. On your screen, you are able to see pictorial representation of two types, two different ways of doing drug design. In 3D QSAR, this is the picture which I have taken from the previous slide. You have already understood the contribution of physicochemical property towards biological activity and all the training sets with biological activity and their contribution and the contribution made by the functional group has been depicted. On contrary, we have 3D QSAR Pharmacophore. So Pharmacophore is a space around which the chemical features are present, which lead to interaction with the receptor and a receptor enzyme and shows biological activity. So it's all about the space, and you are able to see a ligand mapped on that. So Pharmacophore gives a 3D space, the features which are responsible for biological activity and its location. We are able to see the vector also. Vector is the direction towards which the feature should be aligned. So you can know the distance between the features and the vector direction of these features. In the next, this knowledge of drug design can be used for discovering new novel molecule. How we can do? The drug design using 3D QSAR Pharmacophore has a path, has a stages, steps involved in it. So first we have to build a molecule library. Then we have to build 3D QSAR Pharmacophore. Then we have to screen some designed molecules using 3D QSAR Pharmacophore. And then we have to do filtration. After filtration, we have to go for again another filtration with Lipinski rule, Berber's rule, A log P, and then do docking and MD simulation, molecular dynamic simulation. There is some conflict in previous slide, please. There is some conflict in the screening process. Previous slide, please. In the previous slide, let us go back to the previous slide. The discussion we need to make is why Lipinski rule should not be done before. Now we have got the pharmacophore, and based on the pharmacophoric features, we have designed new molecules. Those new molecules should be mapped with our pharmacophore. Better mapping is predicting better activity. When we have designed the novel molecules, why didn't we do Lipinski rule filtration or Berber's rule filtration before? The reason is when we are designing the molecule and we do pharmacophore filtration, we get best mapped molecules. So the best mapped molecule may or may not follow these rules. There is still a possibility of modifying the features and get those best mapped molecules with better fitting values. So if molecular weight, just an example, if molecular weight is more than 500, which will be rejected by Lipinski rule. And we have best mapped molecule. So what we can do, we can reduce the molecular weight and retain the pharmacophoric feature. This way, we have a better strategy of screening with pharmacophore first and then doing screening with Lipinski and Werber's rule. For 3DQSAR pharmacophore, we need molecules with reported activity. What kind of reported activity? Either inhibition constant, IC50 or dissociation constant, that is KD50. So KD or IC50 should be reported. Those molecules are considered for making the library for 3DQSAR pharmacophore. Once our library is there, we need to be sure that there is difference in activity of the molecules. And while collecting the data for the molecules, we need to be sure that the activity has been tested for the same target and assay method is same. I'll give you an example. If the assay is done by flow cytometry or it's done by or fluorescence polarization method, since there is variation in the process of these assays, the IC50 obtained in these two methods would be different. And when after collecting the data, we need to convert the units into same type, all converted to micromolar or nanomolar. The wide range of variation in the structure should be there for dividing into training set and test set. When we divide the molecule into a ratio of test set and training set, there is a conflict that what should be the exact number in training and test set. Ideologically, there should be some difference, definitely. There should be some difference in test set and training set. When we understand that training set is a bigger molecule, bigger set of molecule, then the data generated would be vast, extensive, and more accurate. So we divide the test set and training set in a ratio of 60-40 or 70-30. In some cases, 80-20 ratio has also been considered and successful models have been derived. So 60-40 or 70-30 also works well, it works better. Then we have to divide the set into active set and inactive set based on the reported activity. Based on all these information, we make pharmacophore and pharmacophore generation is done. So after pharmacophore, we screen the molecule and docking is done. I'm just telling you the rough pathway of designing. We'll take it individually later. So after pharmacophore screening, we screen the molecule and then we do docking with the protein structure. So docking is an in silico binding interaction which gives us a docking score. This scoring pattern is based on the previous information of ligand receptor interaction, and it is a dependent value. So based on the previous information, it gives a docking score, and we cannot have single docking score for one molecule to consider it is good or bad. We need to have a standard docking score also. A docking score obtained by the interaction of the receptor with a standard. In some cases where standard drug is not available, then it may be discrete value may be considered, but still it cannot be one molecule's docking score which drives us for any conclusion. We need to have other comparative scores for deciding upon the molecule is good or moderately active. Now, we see that molecular docking can be of different types. On your screen, you see that first picture shows rigid docking, where the protein and ligand, both the structures are rigid. Here, the docking interactions are not so accurate, so it became obsolete. Nowadays, rigid docking is not preferred. Flexible docking has replaced this old system, and in flexible docking, we allow the ligand to be flexible. Why only ligand? Because protein is a big structure with many amino acids and multiple dimensions. So having a flexible dock with flexible protein takes more time and a complex algorithm. In most of the docking process, we keep the ligand flexible and it fits into the binding cavity. Here we compare the docking score with standard, and the standard should be in the same binding cavity. The standard present in the binding cavity should be considered for the docking of the other test molecules. We need to be very careful about selection of binding cavity. Now we'll see that docking consideration for selection of a protein. When we see the protein structure, in RCSP, we get three-dimensional structure. So on your screen, you are able to see ribbon type. It's one of the protein 4N1B, which I have collected, and it is single chain. There can be multiple chains in a protein. For docking, we need only one chain. Which chain, that judgment should be done based on the binding cavity where standard is present, or if there is no standard, we need to find out which cavity is best suited for docking studies. Preferably, we should take human protein, and if we are taking any disease model, we should take protein from the disease model, not from the normal model. So after we have a protein, we need to see that whether it is mutated or not. In the next slide, we will see further more considerations to be meticulous about. You have to do the selection meticulously. Whether mutation is required or not for our study. If disease is caused by mutation, then it is advisable to take mutated protein, whichever mutation leads to whichever disease. So the mutated protein should be present in the binding mutated protein, and mutated amino acid should be present while considering the binding cavity for docking. If there is no effect of mutation on the disease model, then you should not take mutated protein. In case there is no protein structure available, in that case, you have to take the structure where mutation is away from binding cavity. The reason you have to understand the concept why mutation should be away from binding cavity is you will not be able to have the correct binding interaction with the protein when biological activity will be tested. So for the diseases where mutation is not having any effect and you have a protein model with mutation, make sure that it is not present in the binding cavity. The X-ray crystallographic structure should be preferred over NMR structure. When we focus more radiation, X-ray radiation, for better clarity of the protein, some protein loops get damaged. So better resolution structures should be checked about missing loops. Resolution, preferable resolution is less than 2.5 angstrom. Sorry, angstrom is not here in the slide. You need to understand 2.5, the unit is angstrom. So missing loop should be repaired before docking is done. And when we take the protein, it should be ligand bound. Next slide, please. Next slide, please. So ligand bound structure gives us idea about where the binding cavity is. For apoprotein, that is not ligand bound, apoprotein structure. These may be used for the diseases for which there is no ligand, no standard drug, or not, study has not been done. Co-crystallization of ligand and protein has not been done. The one more advantage with ligand bound structure is the protein attains the conformation which it has to after binding of the drug molecule, standard molecule, or your ligand. So that conformation is really important for us to attain after our docking. So ligand bound structure is preferable, definitely preferable over apoprotein. Docking can be used for screening, high throughput virtual screening, and we can do it by we can do docking for interaction study, that is through CDocker. For doing these in silico studies, we need a standard molecule and the evidence of its binding to the cavity. Validation of docking process is one of the important things which students miss and they ignore in their in silico studies. After doing docking, they need to take out the ligand from the binding cavity and do redocking. If the ligand goes and binds to the same cavity, then docking is validated. This is a validation small step which students ignore. It should be definitely included in any kind of in silico study. After docking, the binding interactions are studied for their stability, which is done by MD simulation, molecular dynamics simulation, which checks the stability of ligand in the binding cavity. In the next slide, we see that next step is MD simulation. And again, here we have to do the comparison with the standard. So simulation applies different types of environment which is similar to our physiological environment and checks the stability of ligand in the protein. Let us move on to the case study and in silico study. We'll take an example of KEAP1 inhibitor where we have used all these steps for designing a novel molecule. First of all, we had a test set, training set, then we divided them into based on the activity. Yes, please move on to the next slide. And then we got a pharmacophore. When we generated pharmacophore, we got 10 different pharmacophores. Out of those 10 different pharmacophores, we studied extensively about the distance of pharmacophoric features. You are able to see the round balls. These are pharmacophoric features, vectors, distance, everything. After studying all these, we finalized that the pharmacophore which minimum energy is best one. For that, we studied pharmacophore hypothesis, all the hypothesis, 10 hypotheses, which is shown in the next slide. Pharmacophore hypothesis. And then we have studied the cost analysis. The minimum cost analysis gives the best pharmacophore, and then we got the regression plot done as you see on the screen. So this gives us the idea about active and non-active molecules. So here, after considering the regression plot, pharmacophore hypothesis, cost analysis, we came to a conclusion that the hypothesis 1 is the best pharmacophore. So we have to map our molecule to hypothesis 1. With this strategy in our back of mind, we did comparison of all other, why other 9 pharmacophores are not so good, or should we ignore? There is a question. To get the answer, we compared all the pharmacophores and then we made a cluster of pharmacophores. Anyway, we have generated the pharmacophore which are contributing to biological activity. So we got a cluster pharmacophore. So this cluster pharmacophore gave us idea about query root, which is a skeleton of molecule which can be made based on this, which covers all the pharmacophoric feature and their distance. So query root is the conclusion. Instead of taking one pharmacophore and moving ahead, we compared all pharmacophores, clustered it, got the root structure, and then we designed, based on this, we designed the molecules. We got a 3-ring system idea from this, and based on this 3-ring system structure, we designed our molecules. On your screen, you are able to see the first one is mapped, good map, and the second one is less mapped molecule. So we see that the first figure has more of the features, ring system. Blue, you are able to see mapping with the benzene ring. So here we see that the molecules are mapping better with the pharmacophoric features. In the second picture, we see that only one feature is mapped correctly, other features are very far. So this way screening is done. Then we do docking. So when we do docking, we see that compatibility in the binding cavity of KEAP1 receptor. The KEAP1 receptor structure, which we have taken 4N1B, so 4N1B structure was taken. It has 2.7 angstrom clarity, and we have done docking to the structure obtained from RCSP in the same binding cavity where ligand was found. In the next slide, you will be able to see that 2D interaction with amino acid. So both the solid diagram as well as 2D interaction shows that molecules are fitting good into the binding cavity. We got the binding score. For binding score, it was compared with all the designed molecules which were screened through Pharmacophore, and the best docked molecule was taken further for simulation study. When we see the interactions for one particular ligand, we get multiple conformers and multiple interactions, and we have to select the best energy minimized docking score, and we have to take 2D interaction of that particular conformer of a ligand. So when we see that multiple conformers, suppose 10 conformers generated, the range starts from positive to negative. So negative docking score is the better one. Negative docking score shows more stability. Less energy means more stability. Understand this fact that if energy is high, the molecule will not be stable. So low energy conformer. So the range is obtained. When you see the docking score, from there you have to select and take 2D diagram. Next, the MD simulation of the best docked feature is done. On your screen, you are able to see the graph. Here in the first one, we are able to see that the molecule fits well into the binding cavity, and it is staying well inside the cavity. There are some fluctuations which we see in root mean square RMSF or fluctuation and RMSD, root mean square deviation. So both these plots show that the molecule was fairly stable in the binding cavity. We know that there is no standard or proven drug of KEAP1 inhibition. There is only one molecule which reached to the level of clinical trials where it failed. That is sulforaphane. Sulforaphane doesn't bind to this binding cavity. So we do not have any standard to be compared. Instead, the inbuilt ligand was considered for its comparison, though it is not a standard drug. Inbuilt ligand was considered because there was a co-crystallized structure for that. So we considered that co-crystallized structure for studying and comparing our novel designed molecule. So here, not only this ends the journey, we need to have the activity testing. That is the next level. So in silico journey ends with here. You are able to see on your screen, this is a grid where your molecule, tiny molecule has gone inside the grid, and for which you have already seen the stability graph. It's MD simulation picture. Looks like this. You need to just see that molecule. If you zoom in, you'll be able to find your molecule sitting inside the binding cavity. All amino acids are surrounding, and its stability is justified by RMSF and RMSD. So this ends the in silico journey. So in this discussion, in today's discussion, we have started with SAR, structure activity relationship. Then we moved to QSAR, quantitative structure activity relationship. Then how 3DQSAR became more significant in describing the activity and using as a predictive model. Then QSAR was superseded by 3DQSAR. In 3DQSAR, there are various methods we have used for making pharmacophore, 3DQSAR pharmacophore, generating hypothesis. From hypothesis, we are making cost analysis. Then we are making pharmacophore clusters, comparing pharmacophore, making query root, using this query root for designing the molecule. After designing, we need to do the screening, mapping with the best pharmacophore. After getting that screened molecules, we have to get docking done with the protein, whether it is really binding or not. After getting the best docked molecule, we have to do MD simulation study for checking whether the molecule stays in the cavity. Interaction is one thing, and being stable is one thing. There can be a molecule which shows excellent interaction with the binding cavity and doesn't stay inside for long enough. If it doesn't stay inside the cavity for required duration, it will not be effective for causing the conformational change in protein. It binds, hits the protein and leaves. That cannot be a good molecule. So MD simulation data is required for any docked molecule, and then only we can take the molecule, stable molecule, further for the study in preclinical studies in animal models. So I hope you have enjoyed the journey of QSAR to MD simulation study. You can do this in BioVia Discovery Studio. All the data which I have shown, pictures I have shown, has been done in the BioVia Discovery Studio. My acknowledgement. I need to thank management and faculty of Acharya and BM Reddy College of Pharmacy. I'm an assistant professor in the Department of Pharmaceutical Chemistry. And I would like to thank Atom Technologies, particularly Dr. Sakshi Bharadwaj and Dr. Divya S., for providing me an opportunity to speak with you.
Thank you so much, ma'am. It was very informative session, actually, I would say. I must say that my viewers must have learned, many things from this session. Now it's time to take some questions. We have many questions, actually. So let us take one by one. Okay, the first question is how to calculate molecular descriptors using the Hans analysis.
We have to first get the molecules with biological activity already done. Then we have to align them with based on the descriptors. For example, we have to, for example, when we are doing hydrophobicity, we have to find the contribution of particular functional group towards hydrophobicity. Like previously I have discussed, ethyl group, how much introduction of ethyl group increases hydrophobicity can be calculated by log P value of the obtained molecule, your designed molecule with ethyl group, divided by log P of the standard, the scaffold. That will give the hydrophobicity of the ethyl group. So when you say that will, when you compare for ethyl, methyl, propyl, butyl in a series, you have to calculate. And then the constant, the difference, the constant has to be calculated. That gives a K value and log P value. Log P of your scaffold will be there without the substituent. Suppose it is H, and then you have substituted by ethyl, methyl, propyl, and then you have calculated for each of the substituent. So for each of the physicochemical descriptor, you have to calculate hydrophobicity done, then ionization, same way. If your scaffold, you have to take it with H, what is the ionization constant, and then you will, when you put hydroxyl group, chlorine group, amino group, and then how it is varying, then you will get a constant multiplied, that will be the constant obtained from the average of all those values, where you have got the ionization of your design molecule divided by ionization of the standard structure with H. Same way you have to do with the steric factor. And then you coin the Hans equation. Easier way is, instead of going through all this, you can get it in algorithm-based Hans equation derivation, where you put that substituent and you already get it. Previously, when scientists started, they didn't have in silico studies. So they used to do manually by calculating each of the factor and its contribution.
I don't do, ma'am. Basically, we have some kind of equations to do any kind of, to calculate the activity of your ligand molecule. So basically, this equation we use to calculate the relationship in between the biological activity in terms of with respect to their chemical features. Simplified. So yes, of course, ma'am. Thank you. Next question is: want to know more about the target identification?
So suppose we have, we are considering any of the disease model. In the disease model, which of the receptor, which of the enzyme is more expressed? Recently, my students have done a study where they have taken non-small cell lung cancer. So non-small cell lung cancers can have multiple enzymes overexpressed, multiple of the proteins which are active. There can be some of the transport enzymes active, there can be some synthase enzyme active, there can be some phosphorylase enzyme overactive. But we need to select one particular target which plays role in that particular cancer, which is overexpressed. So they have taken EGFR receptor. So that particular receptor is found to be overexpressed in that particular kind of cancer. So they have identified that receptor. Once they take the receptor in RCSB, you will get multiple numerous structures of EGFR. Then you have to select only human EGFR structures. You can have the checklist where screening can be done. You can remove all of the structure. Only Homo sapiens you have to select for having, because we are not designing any wet molecules now, wet drug. Veterinary drug we are not designing. We are designing our intention is to design for human. So we should take human protein. If it is not available, which is the closest uh, uh, vertebrate or whichever animals, closest animals structure is available, that should be taken. And we have already made the screening pattern. We have to take Homo sapiens, and we have to take from disease model, whether mutation is important, mutated or non-mutated, we have to take. Then clarity should be good. Broken structure we should not take. If X-ray, only X-ray we should take. If not available, then only NMR. Otherwise, NMR is not a consideration for NMR structures are not considered for having, because we will not get clear picture of the protein and we will not get the clear interaction studies. Correct interaction studies cannot be done.
You mention all those criterias. Actually, these things always be a confusion for students, especially when they are going to select those kind of target macromolecules, basically, of course. The next question is, which software to be used for the molecular docking?
See, at this platform, I need to say Discovery Studio, but there are lots of things to be explored outside. I have been using Discovery Studio. All the data which I presented in today's webinar is from Discovery Studio. You can use Discovery Studio.
Okay, the next question is, what are the important study in the cat to confirm the drug for approval by FDA?
Okay. For approval by FDA, we don't need computer-aided drug design. It is only to reduce the cost, time, effort, and animal testing. Its CAD's role is to reduce all these. FDA goes only by your clinical results. Preclinical is, even preclinical, they don't see, only clinical trials result. It will be considered for approval of any drug to become approved or come to the market.
Ultimately, we are reducing our time by using the in silico methods. But nowadays, the government has banned all those animal models what we are using in our wet labs. But in Biodiscovery Studio, we have 12 animal models as such, by the with the approval of FDA and the NDPs, all those authorities. So of course we have those robust animal models to check their toxicities as well in the Biodiscovery Studio to get that FDA approved one candidate finally. So yes, second, we have next question: how to perform 3D QSAR using the computational methods?
You need to collect a set of molecules, a data set, which has same kind of assay done for that activity. For example, you are taking fluorescence polarization. So all the assays done by fluorescence polarization, which has IC50, you collect it. Don't take the same method. Assay method is same, but your result is different. KD. You are taking dissociation constant, KD. So you just be sure that you are taking from the same assay type method, same biological activity, and then the same unit. Collect the data set, and then rest other things you can do in Discovery Studio by dividing interest, test set, training set, providing the activity, and then click, you will get results through the process of 3DQSAR Pharmacophore. You can get it here.
I must add on to this QSAR question actually that in Biodiscovery Studio we have very robust machine learning artificial algorithms to train our models where we have multiple linear regression models, we have partition, partial least square models, we have GFA, that is we call as genetic functional approximation models you can develop. We have the Bayesian models you can develop, where we have the recursive partitioning models you can develop with the different databases. Like if you have the categorical data, you can use those data. If you have the numerical data, you can use those data. If you have only the binary data, of course we can utilize all those databases with the help of all those different kind of methods what we have in our QCR module actually. Yes. Next question. Is it possible to do fragment based drug designing?
Yes, it is possible in Discovery Studio.
Yes, which is very
Yes,
popular nowadays. Fragment
there is
based
an
drug
option.
designing is something
It is
where
an
you.
option.
Yes, yes. Okay, next question is, while doing MD simulation in GROMAX for the protein ligand complex, the ligand restraint is important or not?
Yes, providing the NPT, all the environment is important for having that correct MD simulation result. First, you need to have the best binding, best conformer with best binding pose, and then you have to start with MD simulation. If there is any variation, if one, you will get multiple conformers. From there, the correct conformer has to be selected for doing MD simulation.
Okay. The next question is, how to know the binding site for a new protein target?
For a new apoprotein, you need to have the, in Discovery Studio, there is an option of selecting the binding cavity either through PDB or through Discovery Studio. Automatically generates the cavity. So when we get the binding cavity, there can be multiple sites in Discovery Studio. When you get for PDB, it will be just one where ligand is found. In your case, there is no ligand. There is a generation of binding cavity. The Discovery Studio itself, it shows there are pockets where you can see that study of binding cavity is important before you start doing docking, because it is apoprotein. You need to compare all the possible binding pockets. And based on your study of all those explored new binding pockets, you need to decide upon do docking studies. Do docking studies and compare whether it is enough binding for your molecule or not. It is very difficult for those apoproteins and for molecules to for the diseases where there is no standard drug. It is slightly difficult to get the data, but it is definitely a way to move on.
Yes, we have a very robust, I would say the algorithms to find out those kind of a binding site. Like we have the flood filling algorithms, we have eraser algorithms with us to find out the biology, your binding site, whether it is a apoprotein. Even you have the information about the binding site from the PDB databases or any other databases. You can incorporate all those information, and you can find by selecting those kind of amino acids as well, of course. Do we have any other questions? Okay. So, ma'am.
Thank you. Thank you so much.
Thank you. Thank you so much, ma'am. Do we have any other questions? Okay. Want to start a career in focused on the molecular modeling. So, want to know how to use the similar tools for research in the pharmacology, actually.
So drug design, molecular modeling is not only restricted to medicinal chemistry. It has gone beyond to pharmaceutics, pharmacology. So it has extended. It has extensive use. So it is no more a chemistry department's software. It is used equally in genetics and ecology. So for predictive models and in pharmacological models, which are there for predicting different types of toxicities, lethal lethal doses in rat, mutagenicity, all these things are predicted in Discovery Studio, which I have used for my work.
Where we have a very unique feature in the biodiscovery studio, that is we have the topcat. As I mentioned earlier also, that in topcat we have 12 different animal models to use those kind of toxicity prediction with the different kind of doses like LD50, C50, any other doses kind of means different doses form you can use with the aquatic models, skin models. We have carcinogenic model, mutagenic models, and all are updated to NDA and FDA approvals. Yes. So thank you so much, ma'am. It's a very informative session what we had today. So hope my viewers enjoyed and get they it's more informative. So of course they have got some kind of information. The basic the what the students is lacking in the basics, right? So I think your session is the perfect one to clear all those kind of basics when it comes to the students, right? So thank you so much, ma'am, and thank you everyone for sparing your valuable time for this session. Thank you all. Thank you so much.
It's my pleasure to be here. Thank you so much.